My name is Hannah Bates. I'm the Assistant Director from the Iowa Water Center at Iowa State University, and I will be moderating today's session. Let's see. This webinar is hosted by the Algal Bloom Action Team, of which the Iowa Water Center is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resource Research Institutes, of which the Iowa Water Center is a member, the North Central Region Water Network, and University Extension within each state in the North Central region. Before we get started, we have a few housekeeping items. We will have a Q&A session after our speaker presents today. Please list your questions for our speaker in the Q&A panel, and we will do our best to address all these questions following the presentation. If there is a question already posted in the Q&A panel and you have the same question, please upvote that question and we'll be sure to get those answered, those most popular questions answered during the session. Today's session is being recorded and we will post the Algal Bloom Action Team website following today's session. If you are having technical difficulties or have any questions about the Algal Bloom Action Team, please list those in the chat box and we'll be happy to assist you the best we can. Today's session is the third in the Algal Bloom Action Team's HABS webinar series. These webinars are held bi-monthly and our next one will be held on September 8th. Today's session focuses on filamentous algal blooms and we have a great speaker lined up for you. So without further ado, I'd like to introduce our speaker, Dr. Yvonne Vade-Boncourt, a professor of biological sciences at Wright State University. She will be presenting her newly publicized work on nearshore attached filamentous algal blooms, a new and growing problem. I'll stop sharing now, and Yvonne, you can take it away from here. Oh, hang on, you're muted, and I think we can get that fixed.
Okay, can you hear me now?
Yes.
Sorry about that.
No problem.
Can you see my screen, everybody?
Not yet.
Okay, let me go back. Now do you see my screen?
Yes.
Okay, well, thank you for that introduction and thank you for inviting me to this webinar. Fortuitously, it actually does correspond with the publication of a paper dealing with this new problem. I'm actually an expert in nearshore habitats in lakes. I am not an expert on harmful algal blooms, but nearshore habitats of lakes are being transformed kind of worldwide with this new problem of excessive algal growth. So I'm going to talk to you a bit about that today and just give you some ideas about what we think is happening. As I said, sort of serendipitously, this just was published today about this new problem in Bioscience, and that link below is a link that will allow you to access that article. It is an open access article, and it is written for the general educated audience to inform them about this new problem. So on the left here is a picture of me measuring photosynthesis on rocks at the bottom of Lake Huron. And I'm scuba diving at 5 meters. And most people want their lakes to look like this. They want nice, clear, beautiful lakes where they can see the bottom and there's nothing scary growing under there that will surprise them. That is what is desirable, but because of the way we grow food, especially, because of agriculture and because people build cities around lakes and rivers,
Hello, everyone. Looks like we're having a few technical difficulties. Please stand by and we'll get Yvonne back. Hello, everyone. If you just joined, we are encountering some technical difficulties with our speaker. Hopefully she'll rejoin us back in a minute. Looks like you're back, Yvonne. I'll stop my screen share and let you do screen sharing again.
Thanks. Internet went out. Don't know why. We'll
It
just keep
happens.
going.
Zoom life we're in.
Yeah, that's the world we're in. Okay, do you see my screen again?
Yep, looks great.
I'm not sure where I cut out, but I was talking about what we want to see, nice clear lakes. And essentially what we get is eutrophic lakes, and people who are familiar with HABs should be aware of this, that basically nutrient pollution from the landscape makes the lake water turn green. That actually reduces the amount of light that hits the bottom, and therefore we don't get lots of algae growing on the bottom. Ideally, we never have lots of algae, or we don't have a lot of algae accumulated on the bottom, but eutrophication is a problem that really makes it unlikely that you're going to have a lot of benthic algae. Benthic is associated with the bottom. So what we're seeing now is this new thing where we get this kind of paradoxical event where we have blue clear waters, very oligotrophic lakes, but green bottoms. This is actually the bottom of Lake Michigan with a big filamentous algal bloom that I will get to in a few minutes about its cause. I want to just review a little bit of what habitats I'm talking about. As I said, I'm an expert in sort of littoral zone ecology. Littoral zones are nearshore habitats where enough light reaches the bottom to support photosynthesis. Kind of ironically, they're most extensive in large lakes. Like you expect littoral zone to be important in small lakes, but small lakes tend to be eutrophic. There's no light there. Whereas large lakes tend to have large littoral zones where there's a lot of area that photosynthesis occurs on the bottom. Littoral zones also support the majority of lake biodiversity. So most of the things that people go to lakes for in terms of fish and other things are living in these littoral zones. Unlike this picture that shows all these plants in the littoral zone, most of the photosynthesis that occurs in the littoral zones is by algae that you ideally do not see. So many lakes littoral zones do not have vascular plants in them. They just have slime on the bottom. And that slime happens to be what I study a lot of. Ideally, those algae are eaten and they're not seen. So little critters come along, they consume that algae, then the fish come and consume the little critters. And you never know that there's a lot of algae on the bottom of those lakes. That's the best situation. The other thing about littoral zones is that they're overused and very understudied. So scientists, when they study lakes, it is the way to do it to go out into the middle of the lake and sample from a boat. That means we know a lot about water in lakes, but we don't actually know very much about littoral zones. The public uses the littoral zones extensively. That is the most likely way you are to interact with the lake, whether you're hiking around the lake, whether you're putting your boat in a lake, whether you're fishing, all those things, going swimming, all those things happen at the edge of the lake. So there's this disjunct between what scientists study and what the public interacts with. Now, scientists study the open water for good reason. There's no, I'm not criticizing that, but it has left us with this deficit of knowledge about nearshore habitats. Okay, I'm talking about filamentous algae blooms in lakes, and filamentous algae are a growth form. It's not a taxonomic distinction, okay? So the filamentous growth form is essentially cylindrical or spherical cells grow attached end to end, and that means that they grow long. So over time, they elongate, and you often see them. So we think of them as macroalgae or macroscopic. The algae blooms that I'm going to talk about, the filamentous algae blooms that I'm going to talk about, start out associated with the bottom, which we call benthic, okay? So they start by growing on the bottom, and they may always grow on the bottom, or if things get really lively for them, they'll float to the surface. So if you get high rates of photosynthesis and high biomass, bubbles start to form. So this is on the left here, you see this oscillatorium. This is a blue-green algae. It's photosynthesizing at such a high rate that it's trapping air bubbles, which I think is just super cool. It's just like, wow, that's a lot of growth there. And this is actually a picture of my pond where I have a problem with filamentous algae right now. What happens when you get that so many air bubbles is the whole thing floats up to the top. So often what you'll see is a benthic filamentous algal bloom that somehow goes upward. And that surface scum then is problematic. Often the public see it and don't like it. It can be transported either downslope in a lake or up on shore. Neither one of those are great. So if you get really rapid photosynthesis, you're likely to get these floating scums. There are two major groups of filamentous algae that form these filamentous algal blooms that I'm going to talk about today. One are green algae. Green algae such as Cladophorus, Virogyra, Eleutheryx. Green algae are the progenitors of land plants. They need high light. If you don't have high light, they're not going to grow very well. That's why we're getting these problems in clear lakes. They also like low disturbance. Now, some of them have really strong holdfasts, and so they can form, as you can see on this is a rock in Lake Tahoe with some green algae growing on it. They're very strongly attached to the rock, and they can manage that. But if you had really high wave action, it would tend to rip it off. When they're first establishing, when they're very small, they can get grazed and eaten. But after that, if they're not eaten when they're small and they start elongating, essentially nothing wants to eat them. They're not very tasty. They're really hard to manage, and they're not very high quality food. So basically, if you have grazers there, grazers can prevent them from growing, but once they're established, nothing's really going to eat them. They are not toxic, but they are a nuisance. So if they have big wash-ups on shore, it's very much a nuisance. It's very unsightly. It can make you not want to go to the beach. They start to rot and decompose, which brings in bacteria that we don't want the public exposed to, and they'll stink. They tend to be chartreuse or grass green. I'm saying this because basically it would be good for the public to be able to distinguish between a green algal bloom and a cyanobacteria bloom. So they, as I said, are related to land plants, and they have that bright green color. Some places where we've had really extensive green algal, filamentous algal blooms include Lake Michigan, the eastern basin of Lake Erie, Lake Baikal, Lake Tahoe. You'll see them in farm ditches. You saw them in that little pond of mine that's 3 feet deep. They're very common. The other group of algae that forms problematic filamentous algal blooms are cyanobacteria. These are some of the species that form cyanobacteria, benthic cyanobacterial blooms. Cyanobacteria are amazing organisms. They tolerate high light, they tolerate low light. So they can form very deep in a lake, kind of, right before the light completely disappears, or they can form in a very clear river. They tolerate both high heat and very cold water. You can find them in Antarctica, and you can find them in hot springs in Yellowstone. The ones that form these blooms tend not to be strongly attached to the bottom. So they tend to be sort of slimes that grow over the bottom, and they're very easy to disturb. This is a picture of Lyngbya growing on the bottom of some lake. You can see it's formed an extensive bloom. The lake's not very deep, and it's covering the lake bottom. Lyngbya woolli, actually it's changed its name, but they're also common in Lake Erie. And again, they just are sort of a mess and a nuisance. Their color of cyanobacteria tends to be darker green, so a more forest green color. Some filamentous algal blooms are harmful algal blooms. So some of these cyanobacterial blooms are toxic, and they create a variety of toxins. So Microcoleus, which is formerly Formidium, this is a picture on the right here of Formidium covering rocks in a river in New Zealand. This is becoming very common in New Zealand to have this happen to rivers. Formidium has been around for a long time in low levels, but now suddenly it's just forming these blooms. They form a bunch of different toxins, including antitoxin, Cylindrospermopsin, all these listed here. I'm not gonna bore you with my bad Latin, but essentially they form these toxins, but they form them very patchily. So it's not this bloom of Formidium in New Zealand, it's not necessarily toxic, or you could sample one part of it and it would be toxic, and another part and it would not be. These benthic blooms that have occurred very deep have contaminated some public water supplies in New Zealand. In New Zealand and California and other places, they've caused both pet and livestock deaths. And this is again problematic for the public because the river looks clean, or the water looks clean. It's very clear, but your dog runs in and starts lapping up some water, and it ingests some toxin. The toxins from these blooms are difficult to monitor because they're not distributed in the water column. They're in the cells, which are in this mat that's growing on the bottom. And you'd have to go sample that mat in a bunch of different places. I've shown down here, this is what's called a spat. And I forget I can't remember the acronym, but it's essentially, this is a little embroidery hoop in the Eel River in California. This is an embroidery hoop between mesh, and inside the mesh is a little bag of resin that is sort of continuously sampling and taking up the toxin, if the toxin is there, and then you take that little pack of resin and you go off and analyze it for the toxin. So that is the way that people have come up with to monitor rivers and lakes to see if these toxins are being produced. So those filamentous algal blooms, again, they're occurring more frequently, or we think they're occurring more frequently, and they can be toxic. But again, it's not a habitat that has been sampled a lot, so we don't know. I'm going to go over just a familiar fab that probably lots of people have heard about, especially in the Midwest. Cladophora blooms in the Great Lakes. So Cladophora is a green algae, and it's been facilitated by the invasion of a mussel. So this picture here on the left is of a bunch of zebra or quagga mussels, I'm not sure which, in Lake Michigan. And you can see all this hairy stuff on top of them is algae, it's Cladophora. And the mussels are very kindly giving the Cladophora a place to grow, but they're also fertilizing them. So in general, what happens if you get phosphorus coming into a big lake. So this is what happens in Lake Erie in this panel that I just popped up. A bunch of phosphorus comes into the water column. The algae in the water, say Microcystis, grows really well. What that does is it turns the water green. It doesn't allow, it then causes the light to go away. The light doesn't reach the bottom. The phosphorus gets pumped to the bottom when the algae die, but essentially there's not a lot of room for benthic algae to grow. But if you add this invasive mussel, what happens is the phosphorus comes off the water, the landscape. The phosphorus once again causes phytoplankton to grow in the water column, but this time the mussels, whose job it is and whose sort of goal in life is to filter feed, they filter all that phytoplankton out, and they either poop out or spit out the phosphorus on the bottom of the lake. That phosphorus then provides excellent place for the Cladophora to grow, plus the zebra mussels have made the light climate better, so you essentially have a high light, high nutrient environment that allows these Cladophora to grow. And you get vast areas of, say, Lake Michigan, eastern basin of Lake Erie, where you have these massive amounts of Cladophora because the zebra mussels are making the environment really good for them. So that's a well-known example where we know sort of what's causing the FAB, sort of what its history is relative to the lake use, et cetera. But in a lot of cases, we don't know that. And what we're finding is that FABs have gone global. They're popping up all over. So they have popped up in these lakes, which are very large. These are some of the largest lakes in the world. They're all clear for the most part, so very high water quality and very high water clarity. But now they're popping up in remote lakes in China, away from human habitation, similarly in New Zealand, and in the Rocky Mountain West, and actually also in the Sierra Nevadas, where they're occurring more and more frequently. We don't have great long-term data to show that they're new, but based on what people have been saying, they do appear to be a new phenomenon. Most importantly, these lakes are remote. They're clear, so they have high water clarity and excellent water quality. And for all intents and purposes, they're oligotrophic. So our model of eutrophication just isn't working here because we're getting these massive amounts of algae without the precursors of eutrophication that we would expect. So why is this happening now? One possibility that I'm actually not going to dwell on, but it's possible, is that people have cameras and are sensitized to HABs, and public is noticing them more. And so people are saying, look, this is happening. We're out in lakes and we see this. That could be, but scientists who have studied a bunch of lakes for a long period of time feel that that's not the case. So where we do have long-term data sets, it appears that this is a new phenomenon. Lake Baikal is a great example of that. Lake Baikal has just been utterly transformed by FABs since 2008. So, and we're sure that that's kind of when they started to be a real problem. So I'm not going to say more about that, but we do need to document these better so that we can decide whether these are a new and changing occurrence. Another possibility is that this is just nutrient pollution is getting worse, and FABs are just sort of an early stage of eutrophication. We're adding nutrients to lakes in kind of more and more remote places. And the first thing that happens before we get these phytoplankton blooms is we get benthic algal blooms. So that's a possibility. The insect apocalypse, for those of you who've heard of this, basically that insects are declining owing to our use of pesticides and other things. Insects in aquatic ecosystems are really important. They eat, and what they do is they eat benthic algae, and then fish eat the insects. So they're really an important part of the food web. And it could be that a lot of these are sensitive to pesticides that we're using, and we're reducing their abundance, and therefore the algae are able to grow. And then the sort of elephant in the room is just climate change, that this could be changing how lakes are operating, and that may be promoting these FABs. So I'm going to go through each of those briefly, I'm not going to, as I said, this is a pretty new phenomenon, so we're not sure of any of them, and we're looking into all of them. So are FABs an early warning of eutrophication? This is a graph from a paper just on how in the US, there is a loss of oligotrophic waters. So here they have a cutoff of, on the y-axis of, the percent of stream length with concentrations of phosphorus less than 10 micrograms per liter, which is considered very oligotrophic. Same thing for lakes, where what is the percent of lakes with water quality concentrations or water phosphorus concentrations below 10 micrograms per liter. And you can see that in this relatively young century, they're declining for both lakes and streams. There are just fewer and fewer water bodies with those low levels of phosphorus because phosphorus is being transported to more and more remote areas, and also possibly because climate change and recession of glaciers, et cetera, may be altering how phosphorus gets into the landscape. So if this is just eutrophication, that's problematic because our models of eutrophication are such that we think of the water column. So here, ignore the blue box in this picture for a minute, but just look at the fact that I have light here, percent light, and the amount of chlorophyll in phytoplankton. And the y-axis, such as you see it, is water depth. So up here is the surface of the water, and here's the bottom of the lake. What happens in eutrophication is you get a lot of nutrients in the surface water. This gives you a high phytoplankton concentration, but because lakes are mixed, the concentration doesn't really change with depth that much. That sort of uniformly green water means that light is rapid, light declines very rapidly with depth. There we go, I didn't mean to do that. Okay, so that's what happens with eutrophication. And in terms of where the nutrients are in lakes that are eutrophic, usually the concentrations of nutrients in the water column are quite high. The concentration right at the bottom is even higher. So this is like phosphorus, it's really high right in the mud. But groundwater typically is not necessarily polluted in eutrophic lakes. So groundwater nutrient concentrations are pretty low. So this model of eutrophication doesn't give you FABs because there's no light for them. But what if we increase the concentration of nutrients in the groundwater without increasing the nutrients in the water column? So in Lake Baikal in Russia, groundwater pollution is definitely fueling FABs. So you can see this very coarse substrate, and groundwater basically comes through the bottom of the lake, gets injected into the bottom of the lake. This fuels the growth of algae, but the phosphorus really doesn't even get to the water column because the algae are essentially consuming it all. So if we look at what may be a nutrient pollution problem in lakes where we have FABs, I just explained this graph to you in a fair amount of detail, where you have high nutrient concentrations in the water and low in the groundwater. So this would be the lake water proper. And the groundwater would be, if you dug a hole and looked in the groundwater, that would be the nutrient concentration would be very low. We think in FAB lakes, we have the opposite. So we have really high nutrient concentrations in the groundwater. Those, if it's a groundwater fed lake, that goes up into the water column, but before it can get into the water column, excuse me, it gets taken up by this green algae. So here you have no algae in the water, and then right at the sediment water interface, you have a ton of algae, and very rapidly within that filamentous algal bloom, the light extinguishes. So that could be a way that FABs are forming in particular lakes that have groundwater nutrient pollution. And we're very sure that that's happening in Lake Baikal. And that has really transformed Lake Baikal. This is one of the most biodiverse lakes in the world. It is an amazing lake. It holds 20% of the world's fresh water. The underwater endemic biodiversity is being smothered by these green algae. Sorry, by these green algae. And here, this is Masumi, one of our co-authors. She's standing on the edge of Lake Baikal. This green down here is recent wash up of FABs. And this is last year's, the brown is last year's. You can also see sometimes this on Lake Erie and Lake Michigan when you get big Cladophora blooms and you get a lot of stuff washing up. Okay, so groundwater pollution nutrient pollution could cause FABs. Are declines in grazers promoting FABs? This is like a topic that I really like, so I'm going to give a little bit extra to this, but we don't really know is the answer. But we do know that when you exclude grazers, you get filamentous algae. So here, this is an aquarium in my lab, and I've got an African cichlid here that's essentially eating all the algae that is on the tiles. There, on the other side of the aquarium, there's a wire mesh between them. I just don't have a picture of it, but there's a permeable mesh. On the other side, where the fish can't eat anything, you get this massive amount of filamentous algae growing in the aquarium. All the nutrients that the fish pee and poop out goes and fertilizes the algae. You get lots of algal growth. So what evidence do we have that grazer declines may be promoting FABs? I'm going to show you another picture that just shows in a natural community. These are tiles that were set out in a lake. On the left is a tile that is colonized by a caddisfly that forms these little galleries, these little squiggly lines. It forms those galleries and then it eats all the algae around it. Why are there insects on this tile? It's because they're under trees where the insects oviposit. And so the insects lay eggs, then the adult insects lay eggs in this area of the lake, and the insects grow up, the larval insects go on the tile. In very close to this area, but away from the trees where the insects are laying their eggs, you don't get a nice clean tile with insects, you just get Cladophora blooms. So this is just a natural lake. There's some spatial heterogeneity in terms of where insects are laying their eggs. Where there are insects, you don't get blooms of algae, and where there are no insects, you get lots of Cladophora. And we know this really well, like the susceptibility, the sort of grazers can keep algae down is very well established in both lakes and streams. So there are lots of, agriculture uses a lot of pesticides to kill grazing insects on land. Those same pesticides can kill grazing insects in the water. This is some work by Masumi Yamamuro, published in Science, that shows the use of neonicotinoid pesticides that began in 1993 in Japan and escalated after that in this particular prefecture. These pesticides kill aquatic grazers. They don't have good benthic insect data, but this is zooplankton, so little things in the water column that eat algae. When these are the zooplankton densities before the introduction of pesticides. So you can see that there's lots of zooplankton before they introduce pesticides on the land. Once they introduce pesticides, the zooplankton decline. The same thing happened with insects on the bottom, but instead of having like a 20-year time series, they have before and after, because people don't study benthic insects. But the insects that were being killed were really common ones, like chironomid midges. So like the things that disappeared, they were common. It wasn't like a few species went extinct. It was like really common species were knocked back. So that's a good example of where pesticides affected grazers. But there could be all sorts of other things that are also affecting grazers. We do know that grazers can have a stronger effect than nutrients on filamentous algae. So this is just an experiment by Yang et al. That shows some mesocosms. So these are essentially kind of like big cattle tanks that they replicated. So they have like, at least 4 tanks of each treatment. And it's hard to see, but they're macrophytes. So there are plants in here, and there's water and zooplankton and plankton, etc., and phytoplankton. They have low nutrient treatments here on the top and high nutrient treatments on the bottom. But the big switch of where you see filamentous algae, all this green scum, is in the place where they don't have snails. So if they had snails in there, filamentous algae didn't grow even in the high nutrient treatments. But when they took the snails out, you got this massive amount of filamentous algae. Again, this suggests somewhat of a nutrient effect, but a very strong grazer effect. We really need to target more research and actually monitor aquatic insects in benthic environments in lakes and rivers. We don't do it a lot because it's pretty hard. This is an example from a New Zealand lake, or 2 New Zealand lakes, excuse me, where you have actually a lake that has relatively high water column phosphorus, but it doesn't have, it's clear, it has just a rocky bottom, there's no filamentous algae. A lake very close to it has lower water column phosphorus, but is covered with this fab, this green algal fab. And notice the difference. In the lake that doesn't have the fab, there are 10 times more grazers than in this lake over here that has the fab and basically doesn't have any grazers. And there may be complex feedbacks between fabs and grazers that may make this situation worse. So again, at this point, this is just a hypothesis, but it really does deserve some exploration. So then the other thing is is climate change promoting these changes in lakes. There are all sorts of things that could be happening with climate change. If you've ever been around the Great Lakes in winter, the massive buildup of ice along the edges is really just stunning to see when it occurs. But that mounding of ice, that ice when it forms on lakes, it starts at the edges and it's sort of, we call it anchor ice. It kind of starts at the bottom and then grows up. That action of ice doing that scours the algae and cleans the rocks. But if that's not happening because basically ice isn't forming, then we can get this overwintering of algae. This is actually, this time series down here is in Lake Tahoe. I think it's at 5 meters, which might not have had ice anyways. But basically, you can see that the green algae are perfectly happy to overwinter, and it actually senesces or dies in June. But if ice scour is declining because of climate change, that may be making lake littoral zones more habitable for these filamentous algae. Could be, as I mentioned earlier, retreating glaciers or exposing new phosphorus, or more intense rain events are bringing phosphorus into lakes. You could have changing circulation regimes. Warmer spring temperatures might promote the growth of algae before the growth of invertebrates. And also we could have longer growing seasons that allow these filamentous algae, they're kind of a late stage in succession, longer growing seasons just make it better for them. This is an example of something called a thermal bar. And in large lakes like the Laurentian Great Lakes, the nearshore waters warm up before the offshore waters. And if the nearshore waters are above 4 degrees and the offshore waters are below 4 degrees, those two won't mix. And it could be that nutrients are being trapped there and that's promoting algal growth. Again, we don't know, but that could be something that is at work. Also, we're having increased lake level changes, and we have actually, Al Steinman has shown that this does occur, that if lake levels drop, the stuff at the edge of the lake starts to decompose, and that sort of gets in the water that's going into the lake, and that all those nutrients become available to benthic algae. So all of those are things that could be causing some of these changes. And the reality is that we need to research them more because we just don't know. And as I said, we need to just spend more time studying littoral zones. So what do we do next with this sort of new thing that's happening in lakes? First off, we really, really need to document them. So we need to know where are they occurring, when are they occurring, and how long are they lasting? And I think that we can really engage the public in this, and it's not available now. It's merely a thought in my head, but we would like to do something sort of an iNaturalist.org or something where we can actually allow the public to upload pictures of these, because pictures are pretty powerful in saying where these are occurring. But that's a long way away. That has to be done. We need to monitor more than the surface water. So we need to start monitoring groundwater, and we need to start monitoring benthic algae and benthic invertebrates. And finally, if this is eutrophication, we need to do a couple of things. One, we have to expand our models so that we can kind of work this new sort of type of eutrophication into our models of eutrophication. And we probably need to expand our vocabulary and be very careful about how we talk about this, because I really think it's imperative that we don't confuse the public. Even choosing the name FABs, we went through a lot of iterations to try to figure out what do we want to call these. And we came up with FABs, and we'll see starting today, I think, whether that's an effective moniker for these proliferations. With that, I'd like to thank this international group of scientists who helped me write this paper and whose ideas are contained in this paper. They're a wonderful group. And the various funding agencies, including the National Science Foundation, the Catalyst Fund of New Zealand, and the Tahoe Water Quality kind of network, the Global Water Center at University of Nevada, Reno. And this again is just a reiteration of this link that tells you more about this phenomenon. Thank you so much. Thank you for inviting me. I hope I can answer your questions.
Thank you. And special thanks to you, Yvonne, for sharing your research with us today. As a reminder to everyone on this webinar, our next webinar is scheduled for Wednesday, September 8th at 11 a.m. Central Time. We will now move on to the Q&A section of the webinar. We will draw on initial questions and comments from those submitted during the presentation in the Q&A box. We will do our best to get to all of them. We've got about 18 minutes remaining. Following the Q&A section, please click on the survey link that I will provide in a moment. We appreciate any additional comments or feedback you have for this series. So with that, we can dig into the questions. So question 1, how does phosphorus typically get into groundwater?
That's a good question. So basically, most of our water use from agriculture and sewage and other things sort of slowly pollutes groundwater. The place where it's occurring in Lake Baikal, I think that it's actually fairly direct. It's human sewage that is not adequately managed, and because the sediments are so porous that it very rapidly gets into groundwater. So in some places, like the sediments are really like clay, and it would take a lot to get from surface water down into the groundwater, but that's not always the case. I think that's about as much as I can say about that.
Okay. Next question, is excess nitrogen a driver of filamentous algal blooms as well as phosphorus?
Great question. I would say absolutely. I think that I didn't want to get into it in this particular format, and it's somewhat controversial, but I would say that the green algae that we're seeing are nitrogen hungry. So it could very well be that we are changing the relative availability of nitrogen and phosphorus, and people are working on that. So there's a lot of evidence that if you add nitrogen and phosphorus at the same time, or just nitrogen, you'll actually just shift the taxonomic composition to green algae. And but again, that's not that there. There's a strong possibility that that's the case.
Great, thank you.
They're nitrogen rich. They demand a lot of nitrogen. They don't have a lot of nitrogen, but they need a lot of nitrogen to grow.
Great, thank you. And I see some questions are popping up in the chat box. Please post those in the Q&A box. So that's the button that says Q&A with some talking bubbles at the bottom in the toolbar, and we'll get those questions answered. Next question. What are your thoughts on the impacts of fish stocking on the increase of HABs and FABs?
I have no idea. Well, actually, I would say something in the Mountain West. There are a lot of lakes that were once fishless and then got fish stocked in them. And there is strong evidence, and people are actually researching this. Well, we know what happened when you stock fish like trout in high mountain lakes that didn't have fish before. The fish will eat the invertebrates and things like frogs. And in those lakes where, well, it's interesting, but where they're in lakes in the Sierra Nevadas where fish were stocked, they are more likely to have HABs. And that is probably because the fish ate the insects and the native frogs. Although some of those fish have bullfrogs, and those also have HABs. So they also have invasive bullfrogs also seem to be causing them, but there's a low N on that.
Thank you. We have another question that's very specific to a place. So one participant asked, our lake is a 250-acre warm water impoundment. We are experiencing prolific weed growth. Green slimy algae is covering these expanding weed beds. Are you familiar with that?
So that happens a lot. Actually, I sort of showed a picture that had weeds and then the algae. In general, I didn't hear quite what type of lake this was, but in that situation, it is not uncommon when you get a lot of weeds to then get filamentous algae if you don't have grazers, or if you don't have like wind events or something to mix it up, because these algae don't do well if you knock them off their plants or something. But it isn't uncommon for algae to be associated with macrophytes. On the other hand, if you didn't have the macrophytes in that situation, you'd probably have algae in the water column that would be a lot more problematic. So you can kind of choose your poison, but macrophytes generally prevent excessive phytoplankton, or are associated with if there are macrophytes, you often do not have lots of phytoplankton, and if you have lots of phytoplankton, you often don't have many macrophytes.
Next question, can you speak to any interactions between herbicide treatments for aquatic invasive plant species and FABs or HABs on inland lakes?
I can't. I don't know about that. I think that the whole possibility of herbicides and what they do is, yeah, do herbicides affect algae as well as the plants? What I did just say about macrophytes, about plants, essentially pond weeds. Pond weeds are probably better, like all the research in Europe just suggests really strongly, you'd prefer to have pond weeds than not, even if they annoy you. they do keep water clear. So you end up with higher water quality if you have the pond weeds there.
To what extent are filamentous algae and filamentous bacteria with entrapped algae related?
I don't know. So that's actually sort of microbiome stuff. I think when you're talking, can you clarify with filamentous bacteria, you're not talking about filamentous cyanobacteria, but some other type of bacteria?
Unsure, it's not stated in the question. I think we can have some assumptions there for what.
Yeah, I'm very unsure. I mean, it's actually the bacterial algal interactions, we call that the phycosphere. People are researching that, and it's really interesting, but I don't know anything about it.
Mm-hmm. One participant asks and kind of ponders, wonder if anyone's tried raising and stocking grazing invertebrates, thought they might die off after stocking if pesticides are still high in the system.
Yeah, probably, like with the grazing invertebrates, you probably don't need to, well, who knows what's happening with population densities. It's amazing how fast a water body will be colonized by invertebrates. They're colonized really quickly within a year or two. So if they were there and could be there and could survive, they would be. So most often when we do experiments where we have invertebrates or don't have invertebrates, but I haven't seen anybody who's tried to stock them.
Are you seeing a wide swing in dissolved oxygen levels between photosynthesis and respiration? And if so, could this threaten the survivability of grazers?
Yes, excellent question. So that picture that I showed you of the New Zealand lakes, one of the things we're researching there. So what happens when you have this much algal growth or macrophyte growth? You get huge dramatic swings in oxygen, and that can, and pH, and that can affect actually phosphorus movement from the sediments, but it could also potentially kill the invertebrates at night. So one of the young people who's working on this, Simon Stewart in New Zealand, is sort of setting up experiments to see, is this a positive feedback? If you have lots of green algae, do you get these huge swings in oxygen and then the invertebrates die and then you get more green algae? That's a very distinct possibility.
Could you comment on how your research might be considered in shallow warm water river environments as contrasted to deep freshwater lakes?
I would say that these happen in rivers, and probably they're a little bit like filamentous algae occur in rivers, and we know a little bit more about them. But I'd say there are two things that the algae sort of have different things that would disturb them in a river than in a lake. So the hydrology of the river and the frequency with which you get what we call flushing flows, or really large amounts of water moving through, affect whether or not these develop. And Susie Wood, who works on cyanobacterial blooms in rivers, has found that essentially in rivers in New Zealand, has found that hydrology is really important. Warm rivers are likely to cause, or likely to really be an environment that algae would love. So a warm, shallow river that isn't getting a flushing flow very often might be just a great place for algae to grow. Yeah, it sounds ideal, but there are lots of things that contribute to whether or not we don't find them.
We have a great follow-on question to that. Any research on FABs in large rivers such as the Ohio River?
I don't think so. And I would just say that there isn't a lot of research on FABs, period. I mean, this is like, today's the first day that we're actually introducing this term to the wider audience. I know that we had, yeah, I do not know. Where we have had a lot of research done, the Klamath River in Oregon, and then there's a lot of research on sort of food web effects on Cladophora in the Eel River, which is not in California, which is a large-ish river, but certainly not the size of the Ohio River. Ohio River, I wouldn't think would be a very good environment for it because it's pretty turbid, it's big, so you don't have like shallow highlight environments, and probably the flow wherever it is fast enough to disrupt the growth. What you would likely get, and which they have had, is just regular old cyanobacterial blooms in the water.
Another participant asked, Wisconsin has an excellent citizen monitoring program, and SWIM's website collects the data, SWIMS. What should we ask the Wisconsin DNR for to start collecting data for this issue? For example, we collect dissolved oxygen levels near the shoreline.
That's a tough one. I think that we need to know more about it. In terms of what people typically collect, dissolved oxygen near the shoreline would be terrific in a way, because it probably would give you a good idea of whether the photosynthesis is like going into these big peaks and valleys, but you wouldn't necessarily know what was driving that photosynthesis, whether it was plankton or benthic algae. I really think that actually just documenting what we're seeing is just the logical first step. And as the scientific community gets its head together on this, I think that we'll come up with a better understanding of what we should be doing. And I hope that we can engage a lot of the public and just science professionals in those discussions, because I think it has to go both ways. I think that it's just as important that we hear from people who are dealing with the public what the public are saying to them in order to just sort of understand what should we be doing. Sorry, I can't answer that more forcefully, or more precisely, but I really think that we need to have a conversation about it.
We have about 4 minutes remaining for questions. So is there a link between high water events and FAB blooms on inland lakes, thinking about leaking septic systems?
I would say that is certainly possible. Has somebody made that link? No, not that I know of. So, and yeah, in terms of what lake levels are doing, lake levels are kind of going all over the place, but the extreme weather events could be transporting massive amounts of nutrients to the nearshore areas on a very episodic basis, and that might actually foster filamentous algae.
Are researchers also looking at the role of pH and chloride?
Chloride, I am unsure of. pH, I would say yes, but pH has like all sorts of, pH is a difficult thing because it essentially, when organisms photosynthesize, they increase the pH, and that changes the mobility. Basically, they take all the CO2 out. When you have high rates of photosynthesis, you get big swings in pH, and that changes the mobility of nutrients and metals and all sorts of things. Alternatively, things don't grow real well in very soft water, like not just low pH, but just water without a lot of minerals. So there are also, it's difficult to predict how that would affect filamentous algae. We do know that when we had acidification, lake acidification in the 80s and 90s, one of the symptoms of that were filamentous algae blooms. But the direct link between pH, we don't know what it was.
Okay, I think this will be our last question. In a drought year, would you expect more severe FABs because of nutrient introduction from groundwater? Or would the effect be more from concentration of nutrients and increased water temperature?
I'm a scientist. I would have to say that initially, yes, I might say, sort of without knowing anything else about the system, I might, basically because of higher temperatures and some of the things that this questioner points out, I would say that's a pretty good prediction, but I'd also say I'd have to just know more. There's so many things that go into this. Benthic algae, by the way, is really, really hard to predict what it does, which is why it doesn't get studied a lot. It gets studied much more in rivers because they have to, but it's a tough one.
So I think that'll conclude our questions. Just to show you the popularity of the topic, we still have 22 unanswered questions coming in. So if you have a question in the chat box or the Q&A box and you're dying to have it answered, please reach out to Yvonne and have that question answered. Otherwise, I want to thank you all for joining us today, especially Yvonne for giving us a glimpse at this topic and introducing us to FABs. I myself have never heard of them before, and now I'm excited to share it with everybody. So thank you so much, and please keep an eye out on our website for the recording of this as well as presentation slides. We also have a call out for abstracts for presentations for our annual symposium that we will hold in January 2022. So if you are a HABs researcher also calling in today to have a topic you'd like to share to our audience, please consider looking at our request for presentations and submitting. Otherwise, thank you all for calling in. Thank you, Yvonne, again for speaking, and I hope you all have a lovely rest of the afternoon.